Sexual reproduction and meiosis. So recall that mitosis involves cell division that ensures that both daughter cells are exactly the same as the parent cell. And again, the point of this is to make exact copies of the original cell. Sexual reproduction involves merging of two gametes from two separate individuals, and this allows the genetic information from one person to be mixed with the genetic information from another person, allowing for greater genetic diversity, which is essential for the survival of a species. Haploid and diploid cells. So somatic cells in animals are diploid. So all the cells in your body are somatic cells, except for the ones that produce gametes, sex cells. So the somatic cells are diploid. They have two sets of chromosomes, one of them from each parent. For sexual reproduction, two partners have to produce germ cells, or use the germ cells to produce these haploid sex cells, which we're going to call gametes. These two gametes are going to be haploid. They have half the number of chromosomes, so that when two of them combine, they go back to the full set of chromosomes. So mitosis versus meiosis. The goal of mitosis, again, is to reproduce what is already there, to try to have the exact copy of what was originally there. So the original diploid cell is going to be splitting in two and creating two exact daughter cells. The goal of meiosis is to create 4 gametes. So these are haploid sex cells, and they could be sperm or eggs. They are going to have half the number. So if a diploid cell is 2n, a haploid cell will be 1n, and n represents the number of chromosomes. So the reason for meiosis, again, is if a species is to have sexual reproduction as a method of producing the next generation, those cells that go to make those new individuals, they couldn't be diploid, otherwise every generation would have twice the genetic information of the generation before it. So that wouldn't work out. Even if it did work once, it wouldn't work out for very long. So if two individuals are to combine their genetic information to create the next generation, we can only use half of the original information. So if an individual starts with 46 chromosomes, they're going to have a way of getting that number down to 23 chromosomes. And then another individual with 46 chromosomes would produce cells that also have been halved in terms of their genetic information. So when then those two cells came together, they create an individual that has the same number of chromosomes, same amount of genetic information as the previous generation. So the production of these haploid cells is what meiosis is all about. So do recall that you don't have 46 different chromosomes, fully different chromosomes. I mean, they are different to each other, but they have the same information on them in most cases. So you have 23 pairs of chromosomes that each member of the pair carries the same information. So we call those homologous chromosomes. You have 23 of them from your mother, and you have 23 of them from your father, and the information that you got from your mother is the same as the information you got from your father. And so if we looked at, say, chromosome number 4 here, it's going to have 1 chromosome from your mother and 1 chromosome from your father. It has the same genes on them, but one of them you got the information from your mother, and one of them you got the information from your father. The exception to that would be the sex chromosomes, X and Y. They are going to be different. If you are a male, you have 1 X and 1 Y, and the Y chromosome is going to have different information than the X chromosome. If you're female, though, you have 2 X chromosomes, which essentially carry the same type of information. So each chromosome contains genes on it. So if you have a gene for a particular trait, it'll be found on one of these chromosomes here, of which you have 2 copies of that information. And the location of where those genes are called, or the position on the chromosome is called the locus on the chromosome. So homologous chromosomes, again, you get one from your mother and you get one from your father. And they are going to contain the same information. So imagine we have a pair of homologous chromosomes here, and this would be, say, I don't know, chromosome number 7. So this is chromosome number 7. This is also chromosome number 7. You have 2 of these chromosomes, and they contain the same information but a different version of it. So information as to how the bottom of your ear is structured could be, is going to be on one of your chromosomes. So let's say it's on this one. You're going to get some of that information from your mother, and you're also going to get another copy of that information from your father. They may have different versions of that information. So imagine attached earlobe version is from your mother and free earlobe version is from your father. So that's what we mean by homologous chromosomes. They are the same chromosome with the same types of information on it, but different versions of that information. And the other thing we need to realize is that these chromosomes are made up of chromatids, which are exact copies of each other. So essentially each chromosome, once they've duplicated, there's 2 versions of that information. So you have a copy here and you have a copy here. Both of them are exactly the same. So that's the attached earlobe information from your mother. And then the chromosome you got from your father also has, once the chromosome has duplicated, you have this chromatid and you have this chromatid attached together at the centromere. They are identical copies of that information. So we have a pair of chromosomes, and each chromosome consists of 2 chromatids. The chromatids are exactly copies, exact duplicates of each other. But because you have 2 chromosomes, you have one that you got from your mother and one you got from your father. So essentially, once the chromosomes are duplicated, you have 4 copies of every single gene. Now, 2 of those copies are exactly the same per chromosome. The 2 copies that are from your mother, the 2 copies that are from your father, and this allows these chromosomes later to be split in half, and so we can create actually 4 cells that have the same information in them, though there's the genetic information you got from your father and the genetic information you got from your mother. So we're going to watch how that process happens. The term alleles is the version of the same gene. So we could say the gene in this case is for earlobes, and the allele is whether it is attached earlobe or free earlobes. So homologous pairs of chromosomes, again, you've got these duplicated chromosomes, so one copy and another copy of the exact same information, making up a duplicated chromosome that consists of chromatids attached at the centromere, of which, if this is chromosome number 4, you have 2 versions of that chromosome. And so once it is duplicated, you're going to have another chromatid over here and another chromatid here attached at the centromere, also with a copy of that particular gene on it as well. Because you got one from your mother, let's say this chromosome here, and one from your father, you have 2 versions of that gene. And so eye color genes, to make it simple, or maybe enzyme A, whatever the particular enzyme A is, you're going to get a version from your mother and a version from your father of the same information for the same enzymes. So meiosis occurs in germ cells, and so these are essentially reproductive cells that have the task of producing the sex cells or gametes. And so each of these cells is going to essentially go through 2 divisions, which will give us 4 gametes. And that's how we go from having these 2 sets of double information. You split it twice and you're down to just 1 copy of that information. And that's what we mean by haploid. So each gamete is genetically different, and we'll look at some reasons as to why that is. But 2 divisions, so we're going to start with 46 double-stranded chromosomes. So 23 pairs, and each one of them is double-stranded. So we got 2 chromatids there, or duplicated. That's the 2n number, would be the 46. After the first division, you're going to have 23 of these double-stranded chromosomes. So that's the point where it is haploid, but they're still double-stranded. They still got 2 chromatids, so we go through one more division. And after that second division, you have 23 single-stranded chromosomes, still haploid. So let's figure out how that works. So imagine we had our cell here, and this is our diploid cell, and essentially it has 2 chromosomes in it, 2 different ones. And so let's say the big one's chromosome 1 and the little one's chromosome number 2, of which you have 2 versions of chromosome number 1, that one and this one. And let's say the green one you got from your mother and the blue one you got from your father. They are doubled chromosomes, so they've been duplicated. They consist of one chromatid attached to another chromatid. So you have the chromosome you got from your mother, and you also have the chromosome you got from your father. Or I said that the opposite way. But you have a copy from each of your parents. And this particular cell has another chromosome on it, this chromosome number 2. Same thing, you've got a copy from your father, let's say that's the blue one, and a copy from your mother, which is the green one. After meiosis 1, the chromosome is going to, the chromosomes are essentially going to end up in 2 different cells. So you can imagine the bottom 2 end up in this bottom cell, and the top 2 end up in that top cell. Importantly, this cell has a version of chromosome number 1, and it has a version of chromosome number 2. And this one down here also has a version of chromosome 1 and chromosome number 2. Because if the genes are spread out through the chromosomes, you need to make sure you get a copy of each version of those genes in the resulting cells. If this one up here had 2 copies of chromosome number 1, then it's missing all of the information from chromosome number 2. So that would not work. That individual wouldn't be able to survive without having a copy of chromosome 1 and of chromosome number 2. The second part of meiosis involves taking those duplicated chromosomes and essentially splitting them apart. And so we're going to end up with one of the chromatids here, which once it's split is called a chromosome, and the other one here. So those 2 will be the chromosome number 2. Each cell ends up with a copy of chromosome number 2, and then the same thing happens to chromosome number 1. So each of the resulting gametes, these are called, they have one of the chromosome number 1s and one of those chromosome number 2s. Notice that this is haploid and this is haploid. So what has happened here is diploid means you've got a copy from your mother and from your father. That's essentially what diploid means. So think of it as this individual has 2 pairs of chromosomes. The father version and the mother version make up that pair of chromosomes. That's what diploid means. Haploid means you've got one of the versions. And so this is the father's version for chromosome number 1. And this cell over here has the, oh, I said father, let's say the green is the mother. So it is one of the copies anyways. And this one here has the other one. So haploid means you don't have those 2 different copies anymore. Instead, you have just one version of them, either the mother's version or the father's version. So here's the overview of meiosis 1. And this is essentially making the cells haploid. And in order to do that, you have to separate the chromosome that you got from your mother, separated from the homologous one that you got from your father. And so during this process, and we'll go over it in a little bit more detail, it looks similar to mitosis, but again, in mitosis, you're trying to create exact copies. And if you look at the original one and you look at the final one, these final cells that end up over here, and again, this is only step 1, it's going to continue on. These ones over here are not copies of the original one. So this original one, if we look at it, it's got a, let's say it's got this green one here, which is, let's call that chromosome number 1, and it's got a blue version of chromosome number 1 as well. So it is a diploid cell. It's got 2 versions of the same chromosome, the big one. And then the same with the little one. When you look at the final cell over here, this cell here only has 1 version of that big chromosome. So this is a haploid cell at this point. And this one here, again, the other sister daughter cell, has only 1 version of that big chromosome as well. So it is also haploid. So interphase, again, not really part of meiosis, it's before the division's happening, but importantly, the DNA is being replicated. We go from having the one copy of the chromosome, or a unduplicated chromosome, to having a duplicated one. So two identical copies of each chromosome are attached at the centromere. And so again, that's where we see the sister chromatids attached at the centromere, and they are exact copies. For every chromosome you have, you're going to have in your diploid cells two versions of that. So you have the one from your father and the one from your mother, and if they have a particular gene on them, the one from your father just got duplicated, so they're exact copies of each other. And then you have the same gene on the one you got from your mother, of which on that chromosome they are exact copies of each other, but the trait itself could be slightly different. So if this one's for curly hair that you got from your father, and then maybe the gene from your mother is for straight hair. But both of them would be genes for the structure of the hair. So each copy is a sister chromatid. Again, this is a chromatid, and that is a chromatid joined at the centromere. We call those sister chromatids while they are attached. Early prophase 1, similar to mitosis in that the nucleus is going to dissolve. The asters form, which is associated with the spindle fibers. Centrioles are going to move towards the poles of the cells, very similar to mitosis. Late prophase, there is this process that is unique to meiosis, and this is called synapsis. It occurs when you have the homologous pairs line up beside each other, and they form what's called a tetrad. And so when you have the homologous chromosomes, so that's the version from your mother and the version from your father, they're going to meet, and this thing that they create is called a tetrad. And what happens is they switch parts. And so remember, the genes on each of them are for the same trait, but they're different versions of that trait. So they will switch pieces with each other, and in the end what can happen is that this chromosome that used to be fully made of genes from your father, and this chromosome that used to be made of fully genes from your mother, they've switched parts now. Not all the parts, but just certain ones of the parts. So essentially they're not purely from your father anymore. They're now a mixture, this one here, a mixture of genes from your mother and your father found on the same chromosome. And the same thing's going to happen to both of those homologous versions of that particular chromosome. So this is called the crossing over is what we have at the end here. The points in which it occurs is called the chiasmata. And what this does, and we'll sort of see how this sort of comes together, is that it creates more genetic variability of the cells that are going to be produced. Because if each of these, in the end of it, if each of these chromosomes, now that they're separated, they're chromosomes again, if each of these chromosomes end up into a separate cell, if this synapsis didn't happen, this crossing over didn't happen, both this one and that one would have the exact same information, because remember, they would have come from originally the chromosome that came from your father, which has two chromatids that are identical to each other. So if the crossing over didn't happen, both of those cells would have the exact same information in it. But since the crossing over did happen, one of them has a bit of the genetic information from your mother as well as the genetic information from your father. And this one over here as well, it's not purely genetic information from your mother, it's got a little bit of genetic information from your father in there. So instead of having two types only, you actually end up with 4 different types of cells being produced. If that crossing over didn't happen, you would only get, these two would be exactly the same, and these two would be exactly the same. It would only give you 2 options. Since the crossing over did happen, we actually have 4 possible combinations of traits in those resulting cells. So let's see if we understand some of this terminology. So homologous chromosomes, again, are, let's say we have chromosome number 17. So we're going to have two versions of chromosome number 17. One of these you would have gotten from your father, and I'm going to draw the replicated version, the duplicated version of that. And the other one you would have gotten from your mother. So those are both chromosome number 17. You have a pair of chromosome number 17s, and the state at which I've drawn them is they are made up of duplicated sister chromatids in each one, held together at the centromere. And so a gene that you have on one of them is exactly the same on both sister chromatids within that one, and you have the same trait over here on the version you got from your mother, but you might have a different version of that trait. So same genes encoding the same structures. So let's say it's a particular enzyme that you use for digesting food. You're getting that trait from your father and from your mother, but your mother's version might be slightly different than your father's version, though they do encode for the same trait. So we have our sister chromatids make up the two parts of a chromosome. So those two identical copies within the chromosome. The pair of them, so this guy and that guy, are the homologous chromosomes. A tetrad happens when these chromosomes come together. So if when they line up together, they form the structure once they're sort of attached, ready to exchange pieces. That's called a tetrad, and the synapsis is the result of that process. Chiasmata is the point in which it happens, so the actual specific spot. If this guy trades an arm here with that one, the specific spot in which it is being traded is called the chiasmata, and the event is called the crossing over. So here it is here, the structure as the tetrad. The spot that it happens is called the chiasmata, the location. Synapsis is the meeting of them. Again, remember these two are homologous chromosomes. You have this one and that one with the same traits, different versions of it. Each chromosome is made up of sister chromatids after it's been replicated. And the result, once it has happened, we can refer to that as crossing over event has happened. So metaphase, once we have had all of this crossing over taking place, metaphase is where those chromosomes are going to line up. The homologous pairs are lining up there together, and spindle fibers are going to attach to them, and they're going to they're attaching to the pair of the sister chromatids, and they're actually separating one homologous chromosome away from another homologous chromosome. So the resulting cells are going to end up with one of the two traits. And so this one, let's look at this guy here. So this is one, let's say that's from the father, and this chromosome number 2 is the same homologous pair, but they are being separated from each other. So the resulting cells get one version of the chromosome, either the mother's version or the father's version. But don't forget, those chromosomes might have been mixed up a little bit in a crossing over event, so they won't be the exact version that you got from your mother or the exact version you got from your father because of that crossing over event. So sister chromatids remain together. Their chromosomes are not being pulled apart at this point. So telophase 1, the nuclear envelope is reassembling. We're putting the cells back together. The pairs of sister chromatids are going to be found in each haploid cell. And again, so this resulting cell, once it's fully separated in this resulting cell, these are haploid cells because they only contain one version of each chromosome. Cytokinesis is the actual splitting apart of those two cells. So meiosis 2, this is going to look a little bit more like mitosis in the way that it's happening. So we have, again, prophase, metaphase, anaphase, telophase, but we're dealing with haploid cells as we go through it. So the nucleus of the cells is, again, degrading, so those chromosomes are going to be able to move around. Spindle fibers are going to be forming again, and centrioles moving towards the poles. Metaphase is where they're going to meet up along the equator, and this should look very similar to mitosis, except for we're dealing with a haploid cell. So we, again, only have one version. Let's say this is chromosome number 1. There's only one version per cell, and then there's a one single version of chromosome number 2 in each of the cells as well. So spindle fibers are going to attach at the centromeres again, and these chromatids are going to be pulled apart to opposite ends of the cell, at which point, once they're separated, they again go back to be calling a chromosome. And so you're going to end up with 4 different cells at the end of this. Telophase is going to have the fully splitting of the cells. A nuclear envelope again is going to start reassembling to form the cell, and the chromosomes that have been coiled into that chromosome shape in order to move around can again begin uncoiling so that DNA can be read again. And when it's uncoiled, we call that the chromatin. Spindle fibers disappear as we're sort of ending the replication process. Cytokinesis is going to split the cells fully apart. And again, these are not identical copies of the original cell. They are 4 haploid cells. So the result of meiosis gives you 4 haploid cells. We call these gametes or sex cells. 1 set of chromosomes is found in each cell. And so for humans, it would be 23 single chromosomes. They're not paired anymore. So we have, let's say the big one was called chromosome number 1. There's 1 version of chromosome number 1 here. This cell gets a version of chromosome number 1. This cell gets a version of chromosome number 1, and this cell gets a version of chromosome number 1. But it doesn't have 2 versions like your somatic cells have. Instead, it has 1 version. And notice that the crossing over event has made it so that the version of chromosome number 1 is different for each of the cells. And so if these gametes were used to make a new individual, all of these individuals would get a different combination of the original information that came from the parents. So this individual here would have got the information fully from the father. But this one, whatever information is on chromosome number 1, they've got some information from the father and some information from the mother. And this one here, same thing. They've got some information from the mother and some information from the father. And this one here got all of the information from the mother. So though it's possible we could get 4 individuals from here, those individuals would all be siblings to each other. They would each have their own unique combination of genes because of that crossing over event that happened. Even though the original source of the information was the same, the combination in which ends up in the sex cells is going to be different because of the crossing over. So comparing mitosis to meiosis, the number of divisions, meiosis is twice the number. Again, we're going through 2 sets of divisions, so we end up with 4 cells instead of 2, and those cells are called gametes, and they are not identical to the original one. Again, they're haploid, so that's going to be one big difference, but the meiosis ones are also doing that crossing over event. So not only are they haploid, but they mix up the arrangement of the genes within the chromosomes as well. So the chromosome number, being haploid, is half the original. The meiosis is going to be happening in the germ cells, so they're ones that carry out meiosis and end up producing the sex cells. And again, the point is to produce more genetic variation, which is the advantage to sexual reproduction versus asexual reproduction, or what a somatic cell is going to do is just trying to continue being what it is being. Gamete production is specific, so we're forming 2 types of gamete. We can make sperm or eggs from this particular process of meiosis. So again, we're starting out with diploid cells, and we're forming at the end of it haploid cells. So during sperm formation, the chromosomes, they're going through the process of meiosis, and essentially you end up with 4 viable cells, exactly as meiosis was shown to do. We end up with these 4 different haploid cells being produced, and each one of them can produce a viable sperm. Oogenesis, or egg production, this is going through the same process, but the resulting egg that gets produced, it needs essentially a lot of cellular material. So the genetics in terms of the chromosomes is going to happen exactly the same way. But since the material within the egg itself essentially you need more material there. Instead of all of the cellular structures being divided equally between the 4 of them, essentially the cellular structures end up favoring one of those sex cells being produced. So you end up with one sex cell that ends up with an abundance of the cellular material, so things like the mitochondria and the cytoplasm, and then the other ones essentially don't really have enough of that cellular material to survive. So they become polar bodies, and essentially they are not viable. And so during egg production, only one of the 4 cells that is being produced is going to form a viable haploid gamete. The polar bodies are the name for the ones that are not viable. So there's 2 ways in which meiosis is able to produce variation. The first one we talked about is the crossing over event. Those chromosomes are able to switch pieces, and so the gametes that get produced are going to have a different variety of combinations than the chromosomes that the original cells had. The second one is random assortment. So while the chromosomes are lining up, one homologous chromosome can end up in one cell or the other. And so look at these 2 versions. So look at option A here on the left. So imagine this is option A, and this is option B as to what could happen with these 2 cells. So first off is that maybe by chance, the 2 chromosomes from the father, and again, this is chromosome number 1, and this is chromosome number 2 from the father and from the mother as well. So imagine both of them happen to end up on the same side that are from the father, and both of them from the mother end up on the other side. So when they get pulled apart, so remember they're going to get moved to the other side. When they get pulled apart, you could end up with a cell that has just the chromosomes from the mother, and then one that has just the chromosomes from the father. That could have happened random by chance because of, this is random assortment, because of how it was assorted. Option B randomly could end up with chromosome number 1 on the left side and chromosome number 2 from the father on the left side. So you get the chromosome number 1 from the mother and the chromosome number 2 from the father ending up on the left side of the cell right about when they're about to separate those homologous chromosomes. On the other side, we have the father one combined with the mother. So we could have had this happen just as easily, where you have the resulting cells ending up with a combination, 1 from the mother and chromosome number 2 from the father, and the other one would have the opposite, strictly by chance. And so this is only happening with 2 chromosomes. So you can imagine the combinations possible when you have 23 chromosomes. Just by the way in which they get sorted is going to result in a different combination of gametes being produced. So 2 ways to get variation through gamete production is 1, the crossing over, the exchangement of parts, and 2, the random assortment of those chromosomes while they're being divided during metaphase 1.